Hello students, today let us learn about agents promoting carcinogenesis. Carcinogenesis, also called oncogenesis or tumorigenesis, is the transformation of normal cells to cancer cells forming tumors. Several factors contribute to carcinogenesis and are called carcinogens. The factors that contribute to carcinogenesis may include primary determining factors, secondary determining factors, and favoring factors. The primary factors include chemical substances, the carcinogenic physical agents, and the action of carcinogenic transformation of viruses. The second determining causes are represented by hereditary determination, and only a type of cancer with pure genetic determination is the uni- or bilateral retinoblastoma that affects children. Some types of cancers are also influenced by the hereditary factor. For example, susceptibility to mammary and gastric carcinomas. The favoring causes are risk factors whose occurrence influences the incidence of malignant tumors, and examples include some geographic factors, nutrition factors, sex, age, etc. The factors or risk cofactors involved in carcinogenesis may be exogenous factors or endogenous factors depending upon their origin, and may also include physical, chemical or biological factors. Let us first take up the physical factors. The main physical factors that induce carcinogenesis include different types of non-ionizing and ionizing radiations. Let us see the first type of physical factor, that is the non-ionizing radiations. These are electromagnetic rays with low penetration such as ultraviolet light and infrared radiation. For example, mutagenic and carcinogenic ultraviolet B rays. They are risk factors for eyes and skins. For example, albinism and xeroderma pigmentosum are hereditary factors predisposing to actinic cancer. Next are the directly ionizing radiations. These include electrically charged particles in the form of negative charge or electrons, such as beta rays, positive charged alpha particles. Next are the indirectly ionizing radiations. These are particles without electric charge, such as photons, X-rays and gamma rays. The biological effect of these ionizing radiations depends on several factors including doses, presence or absence of radiosensitizing substances, for example oxygen, and radioprotective substances. Their actions in the cell, including the effects in different parts of the cell such as nucleus, chromosomes, in which case ruptures, deletions and translocations, and on DNA where thymine peroxidation, rupture of the phosphorylated bone at the level of a chain and breakage of one or two filaments occurs, and in the cytoplasm. Next, let us take up the chemical factors. Many clinical and epidemiological observations have established a correlation between the presence of toxic chemical agents and the increased frequency of a certain cancer type in a certain geographical area or within a group or profession. These chemical agents promoting cancer are termed carcinogenic substance. The mechanism of action of the chemical carcinogens group the chemical carcinogens into three main classes. The first one is the direct action or ultimate carcinogens. Such type of carcinogens have the capacity to induce cancer without a previous metabolic activation in the host organism, and examples include nitrosamine, epoxides, ethylamines and beta-propyloctone. Next are the procarcinogens. This group includes most of the chemical carcinogens that are activated after a previous metabolic activation to ultimate carcinogens. Common examples of procarcinogens include aminozoic colorants, aromatic hydrocarbons, aflatoxins, aromatic amines and urethane. Next are the co-carcinogens. These are chemical substances that do not have the capacity to induce cancer when present alone, but they enhance the carcinogenic effect of other substances. That is, they act as promoters in tissues where cancer initiation stage has already started. Chemical carcinogenic substances include three main types: mineral substances, organic substances and mixed substances, for example, tobacco, food carcinogens, etc. First, let us take up mineral chemical substances. Certain mineral chemical substances have the potential to induce tumor growth. Two main examples include arsenic and asbestos. Arsenic induces skin and lung cancer in workers exposed to arsenic, and it also induces hoof cancer in cattle and sheep grazing near plants that utilize arsenic. The next is asbestos, which induces pulmonary and pleural mesotheliomas in miners, industrial workers and animals exposed to asbestos. Other mineral substances include chrome, which induces pulmonary tumor, nickel salts, which induce pulmonary and sinus tumors, and cadmium that induces prostate tumors. Next are the organic substances. Many aromatic and aliphatic organic compounds are also carcinogenic and have the potential to induce cancer. Among aromatic substances, aromatic compounds based on anthracene and phenanthrene nuclei present in pit coal tar induces skin, cervical, lungs, ovarian cancers, etc. Aromatic hydrocarbons such as 7,12-dimethyl benzatracene, which induces skin cancer in the mouse, 3,4-benzopyrene, which is a major air pollutant in industrial smoke and cigarette smoke, 3-methylcolantrine, etc. And amines like 2-naphthylamine, which induces bladder cancer in humans and dogs, and 2-acyl-aminofluorine, which induces hepatic and bladder cancer. Among the aliphatic series, amines or nitrosamines, alkylating agents and plastic packages have been demonstrated to have carcinogenic properties. These compounds are commonly distributed in soil, water, air and foods, and they also have high carcinogenesis potential, especially nitrosamines, which induces cancer in liver, respiratory system and kidneys. Let us now take up the biological factors. Several carcinogenic substances that originate from biological sources are also found in the environment and are taken up by living beings with air, water and food as natural components or contaminants. Such natural carcinogens are derived from plants, microorganisms or animals. First, let us see the carcinogens in plants. Carcinogenic substances in plants are distributed in diverse groups of plants including ferns or pteridophyta, coniferous species or gymnosperms, and the flowering plants or angiosperms. The plant carcinogens include a large variety of structural types such as pyrrolizidin and other heterocyclic alkaloids, alkynyl benzene compounds, furocoumarins, etc. One example of carcinogenic plant is the bracken fern or Pteridium aquilinum, which play a role in the onset of bladder, intestinal and other cancers in some herbivorous species, and the carcinogenic toxins identified in the fern include the main compound tequiloside and others like sicamic acid, pterolactam, petrosius and pterosides, glycosides, camphorol and tannins. Pyrrolizidin alkaloids were the first plant products proved to be carcinogenic, and some other examples of carcinogenic or co-carcinogenic plant factors include reserpine, sanguinarin, nicotine which is present in tobacco, aricolin from chewing tobacco or betel nut, that is Areca catechu and piper beetle, then acronixin present in ashes of Acronixia barreri bark, caffeine, alkynyl benzene compounds found in plants and species such as carrots, banana, parsley, black pepper, sassafras, clove and anise induces cancer with various locations in small rodents. Some phenolic compounds found in plants are also carcinogenic, and examples include tannins, quercetin, rutin, capsaicin, rotenone and gossypol. Next, let us take up the mycotic toxins. Consumption of foods such as corn, peanuts, coconut oil or cotton seeds contaminated with aflatoxins produced by different species of aspergillus have been shown to induce liver cancer. The main aflatoxins identified in contaminated samples include AFB1, AFG1 and to a smaller extent AFM1, AFB2 and AFG2. Some other mycotoxins with carcinogenic potentials are citrine, PR toxin, regulosin, patulin, penicillic acid and G-penicillin, etc. Next, let us take up mushroom toxins. The common edible mushroom Agaricus bisporus contains agaritin and its N-acetylate derivative has been shown to induce pulmonary and blood vessel tumors in rats. Similarly, the extracts of other wild mushrooms like Gyrometra esculenta have proved to be carcinogenic in mice. Next are the streptomyces toxins. Although antibiotics produced from streptomyces species such as actinomycin D, adriamycin, donomycin C, sarcomycin, streptozotocin, azathioprine and bleomycin are commonly used in cancer therapy, their long-term use has been shown to induce cancer. Further, streptozotocin is another toxin carcinogenic for a number of animal species. Next are the animal carcinogens. Many compounds produced by animals are also carcinogenic. Let us now take up some important animal carcinogens. The first one is cantharidin. It is the active principle of the drug called cantharides obtained from insects like Cantharis vesicatoria and other insects from the Myliidae, Omeridae and Staphylinida family. Skin painting with cantharidin in nude mice induce papillomas, squamous cell carcinomas, reticular carcinomas, and or malignant lymphomas. Next are the quinones. 1,4-benzoquinone and its alkyl derivatives produced by insects from the Tenebrionidae and Opilonidae families induce skin tumors in mice when injected subcutaneously and induces pulmonary neoplasms when inhaled. Next are the parasitic trematodes. Certain parasites cause local irritations, fibrous reactions, metaplasia, and finally malignant cancers in men and animals. Examples include Schistosoma hematobium, which induces bladder cancer in humans, Spirocerca lupi, which induces esophageal cancer in the canine species, and Zingylomanioplasticum, causing gastric and tongue cancer in rats. Next are the oncogenic viruses. Viruses are also able to induce tumor in animals, and these oncogenic viruses generally play a role in inducing cancer by transforming cells or causing change in the biochemical, morphological, physiological characteristics of cells. Both DNA and RNA viruses can transform cells, and during the process, the protein coded by the oncogenic genes of a virus may bind to a certain locus of the host cell DNA, which may lead to the activation or inhibition of a gene group that is responsible for the cell transforming character. Virus cancerogens mostly originate from cellular genes of the host, which is excised and incorporated into the virus during transduction. For example, the viral SRC gene is the endogenous cellular SARC genes that is incorporated in the virus when it's separated by excision from the genes of the infected cell. The phenomenon is known as transduction. The mechanism of viral oncogenesis are of two types: the cell deoxyvirus or DNA virus interaction and the cell retrovirus or RNA virus interaction. First, let us take up the cell deoxyvirus or DNA virus interaction. This type of interaction is carried out by DNA virus or deoxyvirus. The mechanism involves the adhesion of the deoxyvirus to the plasma membrane, followed by penetration of the cytoplasm and the nucleus, which is followed by two possibilities, that is, the lytic cycle or the cell transformation cycle. The lytic cycle occurs in homologous cells, which become permissive or permits replication, while the transformation cycle occurs in non-permissive heterologous cells, in which cell transformation is initiated without the replication of cells. Next is the cell retrovirus or RNA virus interaction. This mechanism is carried out by retrovirus or RNA virus. During the first phase, the virus adheres to the plasma membrane and then penetrates the cytoplasm and the nucleus, followed by either cellular transformation or viral production. In the infection stage, the proviral DNA integrated in the cellular genome is duplicated and transmitted vertically in post-mitotic cells of non-permissive or non-virogenic cells, but is transmitted both vertically and horizontally in permissive or virogenic cells. The production of viral particles in the transformed cells may require a helper virus or leukemogenic virus that replicates both the code of its own cell and that of the defective virus in the transformed cell. Oncogenic deoxyriboviruses that induces tumor in animals form a heterogeneous group including pox viruses, adenoviruses, papopaviruses, and herpes viruses. First, let us take up herpes viridae. Examples include the Marek disease virus, which induces malignant lymphomas in chickens, herpes virus Saimiri, which induces reticulosarcomas in chimpanzees, the bladder carcinoma virus, which induces adenocarcinomas in frogs, the pulmonary adenomatosis virus, which induces pulmonary carcinoma in sheep, etc. Next are the Papoviridae. It includes two groups, the papillomaviruses, for example, the bovine papillomavirus inducing cutaneous papillomatosis in cattle, the equine papillomavirus inducing sarcoids in horses, the rabbit papillomavirus inducing soppy papilloma and also carcinomas in domestic rabbits, and papillomavirus which induces sarcomas in mice. The oncogenic retroviridae includes type C oncornavirus and type B oncornavirus. Examples of type C oncornavirus includes the feline leukomogenic virus, which induces lymphosarcoma in cats, the feline sarcoma virus, which induces fibrosarcomas in cats, the bovine leukosis virus, which induces lymphoid leukosis in cattle, and the avian leukosis virus, which induces lymphoid leukosis in chickens. An example of this type B oncornavirus is the mouse mammary tumor virus, which induces adenocarcinoma in mice. Now let us see the conclusion. The agents promoting carcinogenesis includes factors that play a role in inducing tumors. The roles played by the factors may be primary, secondary, or as risk factors. Several physical, chemical, or biological factors contribute to carcinogenesis, and these carcinogens promote tumor formation by different mechanisms. Among the carcinogens, the physical radiations, carcinogenic chemicals, and oncogenic viruses are important agents that promote carcinogenesis. Thank you.